Every genome tells a story. In bacteria, that story can shape human health, agriculture, and the environment. Understanding bacterial genomes is key to tackling antibiotic resistance, tracking outbreaks, and exploring microbial diversity. It gives us the power to predict, prevent, and respond to microbial threats with precision. Join our 4-day hands-on workshop in bacterial genomics, designed to equip you with essential tools and techniques for modern microbial research. Day 1 lays the foundation of bacterial genomics. Explore how next-generation sequencing drives research and diagnostics. Access datasets from NCBI and EMBLENA, and evaluate quality using FastQC. On Day 2, turn raw reads into genomes. Master de novo and reference-based approaches using SPADES, Velvet, BWA, and Bowtie 2, and identify genomic features. Day 3 takes you deeper, from sequence to function. Use PROCA to annotate genes and identify essential genomic elements. Understand the biology hidden in your assembled genome. On the final day, Day 4, it's all about interpretation. Learn to visualize annotation results and extract biological meaning. Create clear, impactful figures and summaries from your genomic data. Whether you're a student, researcher, or new to genomics, this workshop will help you go from sequence to biological insight. Register now. Start your journey into bacterial genomics.